Hello everyone, my name is Al-Khair Adam Khalil Muhammad. I am a PhD student in drug discovery and toxicology at the Department of Biomedical Science, the University of Cape Coast, Ghana. Today I'm thrilled to have this presentation to the virtual conference on chemistry and its application 2024. I'm presenting our study titled In Silico Homology Modeling and Virtual Screening of Plasmodium falciparum acetyl-coenzyme A synthetase for Novel Antimalarial Drug Discovery. This work is a collaboration between me and with Dr. George Gartik Kwanzaa from the Department of Biomedical Science and Dr. Isaac Asiama from the Department of Chemistry, University of Cape Coast, Ghana. Introduction to our study. Malaria remains a major global health issue, especially in Africa. A recent report by the WHO, about 96% of all deaths and 95% of all cases occurred in Africa. Furthermore, the rise of drug-resistant Plasmodium falciparum strains highlights the urgent need for new antimalarial drugs with unique mechanisms of action. Why acetyl-CoA as a target? This enzyme is crucial for parasite metabolism and gene expression, making it a promising drug target. However, limited structural information on this enzyme poses a challenge for structure-based drug design. For that reason, our study utilized homology modeling and virtual screening to identify potential inhibitors. So, the main or the primary aim in our study is to develop novel antimalarial drug targeting this enzyme, ensuring enhanced efficacy and safety. During the recent years, we have known several inhibitors for the Plasmodium falciparum acetyl-coenzyme A synthetase, especially the MMV 693183, the MMV 019721, and the MMV 084978. And the last one is the focus of our study. So, we planned 5 objectives. Number 1, to conduct in silico drug design, docking, and molecular dynamic simulations on analogs of the hit compound MMV 084978. Secondly, we will predict the cytotoxicity of the selected analogs, and then we are going to synthesize, purify, and characterize the high affinity compounds. Number 4, we are going to test the synthesized analogs against the parasite itself and on the target enzyme. And finally, based on the bioactivity testing, we are going to determine the structure-activity relationships. Here is an overview of our methods concerning objective number 1 and objective number 2, which are in silico work. We utilized Schrödinger suite for homology modeling, virtual screening, molecular docking, and molecular dynamic simulations. And we used tools such as Prime in Schrödinger suite, ProCheck, and PROSA for model refinement and validation. Here is an amino acid sequence of the target enzyme as obtained from the UniProt database, and we use it for plus search for suitable homologs and This is the result of the PLAS search. We built a homology model of the target enzyme based on the crystal structure of acetyl-coenzyme A synthetase from Candida albicans with PDB ID 7KDS. Here is a picture of the generated homology model. This model was generated using Maestro, and the binding site from the template was conserved. So as you can see in the right corner of the screen, the co-crystallized ligand, which is a cofactor for the enzyme, which is adenosine monophosphate propyl ester. This slide shows the protein reliability report from Maestro before and after refinement. Some major issues such as steric clashes and unusual B factors were corrected after model refining. So to validate our generated model, we looked at the Ramachandran plot that helps to assess the quality of the protein 3D structure by visualizing the allowed and disallowed conformations of its backbone dihedral angles. So we can see some few outliers on the Ramachandran plots in the disallowed region, but generally the majority of the amino acid residues are within the allowed region. Furthermore, we utilize the PROSA web service to find the z-score of the generated model, and this z-score indicates overall model quality, showing if the input structure score is within the range of native proteins of similar size. And here inside this red circle we can see that our model fit within the range of normal protein with similar size. For the second part of our in silico work, we performed ligand-based virtual screening. We built a library of analogs to the hit compound MMV 084978 by running ligand-based virtual screening. This was done using the Swiss Similarity platform to identify compounds similar to the hit in several databases such as Zinc, Drug Bank, and Swiss Glass. And we use the RPPS web portal to filter the library of compounds based on drug likeness, Lipinski rule of 5, toxicity, and pains alerts. So the outcome is a diverse set of compounds resemble the hit compounds. In the molecular docking, we utilized the GLIDES software from Schrödinger suite to assess binding affinities, followed by molecular dynamic simulations to analyze binding stability. Cytotoxicity prediction was done using the platform from PKCSM and Swiss ADME. And now let's look at the results of our in silico study. We used the hit compound as a query, so we could identify 3,243 compounds with variable similarity to the hit compound. Some compounds are duplicated, so we remove the duplicates, so we ended up with 2,497 molecules. We use the ligprep function in Schrödinger suite to generate 10,707 species by creating stereoisomers, tautomers, and all ionized species. Then we filter those compounds, resulting in 5,268 molecules passing the filter we prepared. This filter considers certain criteria like the molecular weight, the number of rotatable bonds, and the number of heavy atoms, so we can have a set of compounds with specified criteria. Finally, we utilize the FAF drugs for service from the RPPS portal to eliminate molecules with potential admitox related issues, resulting in 3,620 molecules passing this filter. So this final library of more than 3,000 molecules are docked into the Homology model of the target enzyme. Okay, now let's look at the molecular docking results. Before docking the library of analogs, we validated the docking protocol. The co-crystallized ligand was redocked into the binding site. 14 different poses were obtained, and the RMSD of the best dock pose had an RMSD of 0.92, as shown in the table. And in the picture at the left side, we can see that the co-crystallized ligand is shown in a cyan color and the redocked ligand in a yellow color. Both structures are superimposed and they are not different so much from each other in the binding poses. So this interpreted as the docking protocol was able to reproduce the same binding pose of the co-crystallized ligand, and hence the protocol is valid. Therefore, we proceeded to the molecular docking. Out of the 3,620 docked molecules, 2,717 molecules showed favorable interactions. We constructed an interaction fingerprint and found that all docked molecules have certain favorable interactions with key residues within the binding site. These are indicated in the graph. The most common residues were glycine 679, tyrosine 705, tryptophan 706, aspartate 794, arginine 809, asparaginine 8115, and arginine 820. These are the most important binding residues within the binding site. We selected the top 10 compounds based on several criteria, mainly the docking score, which should be higher than the hit compound, the synthetic accessibility, predicted admitox properties, and physical properties. Here is a list of the selected compounds to be considered for the next stage of our study. These compounds are currently being synthesized in order to test them in vitro for bioactivity, including the hit compound itself. We had determined the ADMET properties of the selected compounds. ADMET stands for absorption, distribution, metabolism, excretion. The following slides present the ADMET prediction of the top compounds, highlighting key properties like molecular weights, log p-value, hydrogen bond acceptors and donors, synthetic accessibility, and more. Here we have the solubility, the GI absorption, the blood-brain barrier permeability, the permeability glycoprotein substrate potential, and the capability of those compounds to inhibit this carrier. Here in this slide we have the metabolism characteristics of those molecules, either as substrate or inhibitors to variety of cytochrome P450 enzymes. And here we have the toxicity profile of all the top 10 compounds. Some of them have slight issues in toxicity or mutagenicity, but the majority of them have no issues. Then we did the MD simulations for the top 10 compounds. We conducted the MD simulations for 50 nanoseconds to further evaluate the stability and dynamics of the top scoring compounds in complex with the enzyme. This helps us identify compounds that maintain stable interactions over time. These slides show the root mean square deviation or RMSD's value of the ligands during the MD simulations, indicating their stability and flexibility within the binding site. Here we have the hit compound itself. In the next slide we have compound 555, which show higher stability compared to the hit compounds, and some of the bonds are maintained for the entire time of the simulation time. Here we have other compound, the third, fourth compound, the fifth compound, this is the sixth compound, this is the seventh compound, and this is the compounds of these RMSD values for all of the compounds combined together. And as you can see here, no compound reach 2 RMSD value, so this is generally considered high degree of stability of those ligands within the binding site. The following slides show the ligand contacts with the binding site residues as obtained from the MD simulations. These contacts are hydrogen bonds, water bridge, hydrophobic interactions, and pi-pi stacking, with numbers indicating the percent of simulation time each compound or each bond was maintained during that time. Some contacts show more than 100%, indicating more than one interaction with the same residue over the simulation time, which was 50 nanoseconds. For example, here we have ARG 820 have interaction with this oxygen for 180%. That means it has more than one bond over the simulation time. Here we have compounds 14 and 11, and these compounds 17 and 8. So in conclusion, our study successfully generated a reliable homology model of Plasmodium falciparum acetyl-CoA enzyme A synthetase, which allowed us to perform virtual screening and identify promising heat compounds. The synthesis of these heat compounds is currently underway for in vitro testing and further optimization studies. This is a roadmap informatics, a visual roadmap of the research process, summarizing the key steps and highlighting future directions. Here is our references. And finally, I would like to express my sincere gratitude to my research team and collaborators for their invaluable support in this project. I would like also to thank the VCCA organizing committee for the opportunity to share our work at this important conference, and thank you.